Well, let's talk a little bit about biology and some things related to that. A lot of things, perhaps more things than biology itself. Yeah. And Angelo, we know each other since 2000 something, 6, 7, and Angelo is a very how can I say, clever guy. Huh?
Thank you.
And a very robust researcher in terms of producing knowledge and advertising it and communicating. So he's leading groups of people in Brazil. He was one of the authors of the book we launched on Monday. And the way he is working is a way that unites people. And that's why it's a pleasure to have you here as a colleague, as a friend, not as a couple, as I said in the first day, but as a partner, okay? Yeah.
Because now we are
couple,
we are live, you know.
We are
live now, right?
Yeah.
Yeah.
So, Angelo Bernardino with you.
Thank you, Alex. Thank you. So my first proposal here in this afternoon is to take a picture of this audience. Fantastic. It's the first time I give a class to people from 23 countries, and I'm going to post it in Twitter. Are you okay with that? Yeah. Okay. Okay, say hi. Woo!
No. Vem cá.
Nice.
Only this part of my head.
Yeah.
Okay.
Very nice.
What's your tweet for the
I have to look at him. I'm not that. I'll give it to you later. Okay, guys. Thank you. I have to thank Alex for inviting me. It's a pleasure to be here during this whole week. I was very pleased to see all the talks and the posters outside. I'm sorry for those that I haven't made to the posters. I mean, I promise that if you come to me, I'll happily look into your work and make some comments if you wish. So today I'm going to talk. Let me just record time here so I don't speak too much. So I'm going to talk a little bit about marine biodiversity. It's, of course, a theme that I could speak for long hours here, so I understand that most of you, or maybe a lot of you, are not biologists or are not oceanographers, so I'm going to try to bring attention to you of some important oceanographic patterns, phenomena, and link it to marine biodiversity, its processes, and things and services that we might lose with climate change impacts. It's a really long theme, so I'm going to try to bring your attention to some important stuff so you can also look for yourself at some further information. So a little bit of my background. I'm a biologist, so I came from the countryside of Brazil, it's a town, Belo Horizonte. It's a long way from São Paulo here where we are, so I came to São Paulo after my biology course and did my master's and my PhD here at the University of São Paulo. And I was telling to some people, so the first time I got in contact with marine ecology was after I graduated in biology, so it's never too late, right? So I learned most of the things that I know, the foundations of my oceanographic background here during my master's and my PhD. And I did a little bit of my PhD in University of Hawaii in the US, good place to go, right, to study. And I studied on my master's and on my PhD, I studied basically deep sea ecology. In my master's, I studied polar ecosystems, Antarctic specifically, so it was really good to give a background. I was studying the Antarctic continental shelf, looking at benthic ecology, and the Antarctic continental shelf, many of you might know, is a deep continental shelf because of the weight of the continent, right, a lot of ice. So it's about 700 meters depth, whereas most of other continental shelves have like 200, 100 meters depth. So I started to do a deep sea ecology back then in my master's, and then I moved to the PhD working with chemosynthetic ecosystems in the deep sea floor. I worked with a famous guy, Craig Smith, from the University of Hawaii, was the guy that discovered the whale falls, right? It's a fantastic chemosynthetic ecosystems that starts to develop after the whales die. And when they die in the open ocean, they sink to the bottom, and then you have a ecological succession on that whale, and a lot of very special organisms colonize and eat the whale. So I worked a lot of deep sea ecology during my study years. And after I finished my PhD, I moved up a little bit to Victoria, where I am right now. And I've been a professor since 2010, about 8 years. And there I kept studying a little bit of deep sea ecology, and mostly moved to the coast due to funding limitations, and working with estuaries and mangroves and some coastal processes, and always trying to relate biodiversity with impacts and loss of ecosystem services and stuff. So that's quickly my background, and I'm very happy to, like I said, to show you some of these biodiversity patterns today. So I'm going to talk briefly in this first half of my class today about marine biodiversity, showing some important global general patterns and processes, showing how our oceans are heterogeneous. A lot of people talked about this before me, and the importance of primary production, and a brief review of processes in the oceans, and how these affect biodiversity. And I'm going to show at the end of this first half some cases of how we can apply biodiversity or ecological data to conservation, which we always want to do it, right? So, and then at the second half, I'm going to talk specifically about climate change, its impacts. I'm going to wrap it up, and we can have some time for questions and debate. Okay. So just a quick review. I think most of you know by this time that the oceans is the biggest part of our planet, right? So we have about 70% of Earth is covered by oceans, and like people said before me, 90% of the habitat on Earth is oceans, right? So that's a pretty amazing number, I think. And the mean depth of the oceans is 3,800 meters, which is basically deep sea, right? Very deep sea. So we consider, for definitions, we do divide these areas into habitats or regions. Like I said, we call the continental shelf up to the first 200 meters of water. Usually, there are over the extension of shelves on the continents, close to the continents, and under influence of the continents, like Professor Drude mentioned yesterday, gave a talk about this yesterday. So I'm not going to talk a lot about the shelves. Then you have the continental slope and the bottom and the abyssal plains, which is most of the oceans. So it's pretty interesting that most of Earth is deep sea. And here you have a map that you can have a better idea. So everything in blue in that map is below 3,000 meters. And that gives you a really good impression, I think, of how much deep sea we have in the oceans, right? So a lot of the of Earth is under really permanent dark conditions, cold conditions, waters below 2 degrees Celsius or even lower, and more importantly is below is food limited, right? We don't have light, we don't have local primary production. And so these animals, they must cope with high pressure, and these guys the animals must cope with all these limitations for their lives. And at the same time, we do have, like I said, a lot of interesting habitats in the bottom. I'm not going to talk about them specifically, but you do have different conditions. Like I said a little bit earlier, you have chemosynthetic habitats, you have mudslides, you have organic falls like a whale carcasses going to the bottom. So you have a very rich number of habitats, which makes special, brings special hosts, special biodiversity. And it's interesting, I think you might have heard about it. We know better, we have a better map of Venus than of the bottom of the oceans. Did you know that? So we have already, we have in more detail the map of the surface of our Mars. I'm sorry, not Venus, Mars. We have a better map of Mars, of the surface of Mars, than of the bottom of the ocean. So most of the abyssal plains are completely unmapped. And there's currently now a global effort to map the whole abyssal plains, the whole bottom of the ocean. So, and it's happening now in almost 2020. So it's amazing that how much of the bottom we don't have yet even mapped. We have an idea of the depths because of satellites, right? So satellites, they can, go over and have an idea of the depth, but we don't have the maps in detail. So we don't know a lot about the habitats that lie in most of the oceans of our planet. So just a quick review, Professor Edmund just said about it, a lot about physical processes, right? So basically we do have in the most of the surface oceans, where there's primary productivity, where sunlight heats water, you have warm waters on the surface, and on the poles, either southern or northern poles, you have cold water sinking to the thermohaline circulation. We just had 3 hours of that, so I'm not going to repeat it. But the important thing for that is that when the bottom water gets formed in the poles, it sinks, and then you have a pretty much similar pattern. And these two plots here, the left one is the temperature at 2,000 meters depth, and the different colors shows you different temperatures in degrees Celsius. So the darker blue is colder, and the red is warmer. So, and then you see by color similarity, right? You see here that's the North Atlantic deep water that flows down in the Atlantic, and in the Pacific you have a little bit more warm waters, right? It mixes more with warm waters, and then it gets, as it goes through the bottom of the ocean, the water gets a little bit warmer. And that's, like I said, 2,000 meters. On the right side, you see that's the water temperature at 35,000 meters, which is basically the abyssal plains. And then you see, if you compare, we have a very different temperature between the Atlantic and the Pacific, right, and Indian oceans, which is pretty interesting, right? So even though these differences are small, like I said, the dark blue is about from -1 to 1, and the light blues are between 1 and 2 degrees Celsius. So it's pretty cold anyway. But the important thing to know here is that temperature has a big influence, not only in organisms, but on the chemistry and on the mixing and on the process, biological process at the bottom, right, and in organisms. And with the temperature also, you have an idea, like Professor Edmund said a little bit earlier, you have an idea that you have to think about that water is being transported, and like he said, you have animals, you have larvae and microbes, a lot of things being transported with it along the deep ocean basins. And also, I'm going to show oxygen later, I think. So, so the temperature, salinity, and oxygen and nutrients are really important to define the major oceanic basins. So these water masses, they really define some biogeographic regions in the global ocean, and I'm going to show that a little bit later today. So that's really interesting to think about. And just one more point here is that basically you see that when you get to really deep waters, there's basically no physical barriers to dispersal, right? So it's pretty much like, no big abyssal plains with long extensions of water, and except of course for the mid-ocean ridges where you can see here where the tectonic plates meet each other. Okay, so like I said, the ocean, we go from the surface of the ocean to the bottom of it, it's of course not homogeneous like you just heard. So we do have at the surface of the oceans, and this is a profile of nitrate, right? Nitrate is an important nutrient for plankton, for microorganisms. And we have a depletion, this is a typical profile, right? It changes a little bit from place to place. But anyway, what is typical here in the open ocean, you have a sunlit layer, mixed layer, like you've heard, of productivity, and the plankton sucks up these nutrients and produce organic matter, right? Photosynthesis. And so, and as the photosynthesis, as the ocean gets deeper, the nutrients, they become recycled, right? So this is what in the inset here, you see nitrate is being converted to particulate nitrogen, and that's organic matter. And that particulate nitrogen, it sinks to the bottom, a little bit gets recycled along the water column, but then it keeps up adding up at the bottom because you don't have a lot of production in the bottom, right? It's dark. So the nutrient layer, like they said, the nutrient line here is typically that. It's depleted in the surface because of primary production, and it gets a little bit, it increases with depth. And as you might expect at the bottom, so you have the opposite processes, right? So we have the particulate nitrogen being converted in dissolved organic matter, degraded by microbes, and getting recycled again into nutrients, into new nutrients. So when you have like physical processes that bring this water up to the surface, like upwelling regions and other, western boundary currents, you do have replenishment of nutrients, and you have new primary production, right? And this is basically controlled by temperature gradients, right? Like Adam just said. So we do have the differences in gradients. You have this stratification between the surface and the bottom. And when this stratification decreases the strength, you have new production coming in, right? Or regenerated production on the surface. And when you look at the oxygen, it's a little bit the opposite, right? So oxygen gets mixed in the surface of the oceans, and it gets depleted in the bottom because of course, respiration, right? So we have a balance between photosynthesis and respiration, and there's a lot of microbes respiring organic matter, decomposing organic matter in the mid-ocean layers. And this is a really cool plot. I first became in contact with it. It's a review by DeLong and Carl. And I'm sorry, all the papers that I got, you can Google it and you can find it easily after you look at it. So when I was a student back in, I think it was 2008, I took a course, a microbial oceanography course, which is a fantastic new word that I'm not sure if many of you know. And I was really impressed to learn at that time that how many different bugs are there in the ocean waters. It's amazing. Any of you know how many bacteria or viruses are there in 1 cc of water? 1 ml. Any number? Million? Who thinks a million bugs? No? 10 million? No? 100 million? No? A billion? Well, it's a billion, right? In 1 ml of seawater, you have 1 billion bacteria. If you get all the bacteria, the viruses, and their archaea, which is different from bacteria, you have 10 to 12, so it's about 100 billion bugs in 1 ml of water, of seawater. That is amazing, isn't it? So imagine that most of the ocean has like billions and billions, hundreds of billions, trillions of bugs, of viruses, archaea, and bacteria of all kinds, of all types of metabolism, of all types of production of proteins and everything else, that are basically transforming organic matter produced on the surface until it sinks to the bottom. And it's amazing. Some of them, they make photosynthesis, which is not photosynthesis, chemiosynthesis, and other process, and absolutely no sunlight. And they use all kinds of dissolved organic matter that I just mentioned, which comes from plankton poops, or rests of small organisms. So they recycle everything into new energy, and sometimes they are really important trophic link to the biodiversity of the pelagic zone, of the mesopelagic zone, because they absorb dissolved organic matter, which is not very useful for metazoans, right? And so metazoans, like sponges or like other filter feeders, can eat these bugs and so use that energy. So it's amazing that, so this is the classic on the left, it's the classic food web, food chain that I had learned when I was a student, which is like, well, okay, we have primary production, some of that algae, diatoms and the large algae, right? They are really large compared to the phytoplankton, right? And when you have like, you have the algae, and then that algae is eaten by zooplankton, and the zooplankton gets eaten by fish or by other invertebrates like benthic animals and stuff. So this is the classic food chain as we know, and it happens as well. Like I said, in regions where we have really strong primary production, like in Antarctica, you have large algae, and you don't have a lot of nutrient limiting layers. You have a lot of nutrients, you have adequate conditions. You do have large algae producing a lot of organic matter and getting eaten by large zooplankton, like the phazids, where the whales go and eat them. So this is the the short food chain, right? And whereas in the most of the oceans, in the oligotrophic gyres, in the middle of the oceanic bases, where you don't have a lot of nutrients, a lot of the primary production, which is low, is really recycled before something gets sunk, something arrives to the bottom of the oceans. So this is like a really interesting aspect of the microbial world, right? Which they call like microbial loop, right? So there's a huge diversity of microbes, and a lot of them that are still not known because you cannot cultivate them in the laboratory. People go there, you get a bunch of water at 3,000 meters depth, but you cannot cultivate them in the laboratory. They don't survive, right? There's light in the laboratory, there's not a lot of pressure. So it's amazing, and they only started to discover this hidden biodiversity by sequencing, by DNA. So they know, they get the DNA, they sequence, and they say, oops, I've never seen this gene before, right? And the most abundant plankton organism, actually bacteria, the most abundant bacteria in the world, it's about 25% of the numbers, was only discovered about maybe 10 years ago. So it was about the time of that paper, they just had discovered SAR-11. Maybe some of you have heard of it. And SAR-11 is a bacteria that was discovered because of metagenomics, techniques, new genetic techniques, right? So it's amazing, right? It's amazing. It's an amazing microbial world. And which is more interesting, 90% of the biomass on Earth is composed by microbes. That shit is really cool, isn't it? Oh, I'm not supposed to say shit here. But anyway, it's really cool, right? I think it's really cool. So microbes make up the bulk biomass in the world, in the planet, which is really cool. Anyway, so like I said, when we have these two types of general, right, trophic food webs, where on the left, we have the large plankton PL, eating, absorbing nutrients, and then you have the higher trophic levels feeding on the plankton. So you have a short food chain. There's a more energy efficiency being transferred here, right? Whereas in the middle of the ocean basins, the phytoplankton, the viruses and everything else, you have nitrate or ammonia or whatever being recycled, and then you have a really small plankton, and then you have a long food chain before this particulate organic carbon gets used by an animal or a metazoan in the bottom of the ocean. So these differences in pelagic food chains, like I said, they have a very important result in the amount of food that is exported to the bottom of the oceans, right? So we need to understand what happens to the animals and the biodiversity that depends on this exported organic production from the surface layers. And this is called the benthic pelagic coupling, right? So that's the biological pump. That's famous, right? So what you have is, like I said, you have a sunlit layer, usually with nutrients. You have algae and phytoplankton producing organic matter. A little bit of that organic matter gets exported, which means it is not recycled within the sunlit layers. And then you have sinking, like these dots here, and then you have sinking material, organic material, going to the bottom. And about at 3,000 meters, about only 5% of the net primary production gets there. So it's not a lot of food. It's very little actually, right? And then with this production, you have all the happy bentos feeding on it on the surface of the mud, of the bottom of the seafloor. And it's important, like you can imagine, like if you have a lot of energy, you just came out from lunch, so you had a lot of stroganoff, and so you get energy, and then you get active, you do everything you need, right? But at areas where you only eat like a piece of bread or something less nutrient, with less nutrients, you don't have a lot of people in there to feed, right? So that the amount of POC flux to the bottom of the oceans controls is really directly related to the benthic biomass, abundance, and body size, right? So usually, right, because of food limitation, as you increase depth in the oceans, the biomass decreases, of course, the abundance of animals decrease, and the body size decreases. So you have a fundamental change on the biology of organisms from the shallower oceans to the bottom of the deep sea. And this is really important. I'm going to show you that. Why that? Because these parameters are important for processes in the most of the globe, right? Remember, over 60% is deep sea. So it means that POC flux, the primary productivity, and the export, the biological pump, is really important, not only for feeding organisms, and that includes fish, of course, which are way up there and until the bottom, but the amount of food that is exported is also important for the recycling and the bioturbation and the burial and sequestering carbon on the oceans and in most of the globe, right? And that's important. So, for some time ago, for at least 20 years, and there are papers much older than this about the subject, I just got the new stuff, people have realized that this is a review by Rex et al., then this is like the biomass, right? So this is biomass and this is depth, increasing depth. So you see that for the blue line, the green line, and the red line, which are biomasses of macrofauna, which are organisms about half centimeter in size, worms mostly, and the megafauna, which includes sea stars and urchins and stuff like that, and myofauna, which are the nematodes, which is most of it. So the biomass of them, you see, with increasing depth, decreases markedly, right? So that's a food limitation effect, right? Like I said, there's less food as you increase depth. And this black line here is the microbes, right? So there's not a lot of effects on the food limitation on the microbes, which is really cool, right? So they don't care a lot about what's going on with the food limitation. So that's why one of the reasons we have a lot of microbe biomass in the oceans. And when you look at the gradient of POC flux in some areas, so that's in grams per centimeter per year. So you're talking about 2 grams. Some areas have like half a gram of organic matter per centimeter square per year, right? So the position rates of food in the oligotrophic basins it's at a rate of about 1 centimeter at every 1,000 years, whereas in mangroves, you heard about mangroves yesterday, is about it can be up to 5 centimeters a year. So it's very, very food limited. So, and then this POC flux controls activities of animals at the seafloor. Not only biomass, but activity, bioturbation. So when you compare similar areas with similar depth, you get more activity, more animal bioturbation, which means like includes sediment oxygenation, includes remineralization, includes nutrient efflux from the bottom, from the sediments to the water at the sediment-water interface. You get more activity in areas where you have more food, right? So that's really interesting. So like I said, and respiration, of course, right? Respiration is the purple light. So everything is really, really correlated with the amount of food. And so this is really important. So basically, the bottom line here is that production at the surface, at the ocean surface, controls not only the amount of animals and densities, and we're going to see some relationships with diversity of the bottom bentos, of the deep sea bentos, but also controls the process, the speed of processes occurring at most of the seafloor and the oceans, right? And this is important. So, like I said, so when we look, and this is a paper, this is primary production, and actually this is export photosynthetic carbon. So the red areas, the hotter, right? The hotter areas are where most of the production occurs or is exported. And there's no surprise in that, right? So most of the production in our oceans occurs on the continental margins, right? And that's where the fisheries are. So there's a direct correlation. It makes sense, right? We get most of the fish on the margins. Here's the Peruvian upwelling. We have some the Patagonian shelf margins, very productive, African and others Alaskan. And so where you have a lot of fish, you have a lot of production going on. So that's, like I said, it's a trophic food chain, right? It's a direct link. And when you compare this with this map here, and this map here is the benthic biomass. And you can see the color patterns match a little bit, right? You have the equatorial upwelling here, you have a little bit biomass here. So it's a direct link, right? So that's globally, right? And that's been reviewed recently, showing that, okay, the production is really linked to benthic biomass, and that's linked to production and fisheries production. So that's pretty good to look to predict where the fisheries are going to be and where most of the bioturbation and all the process are going on the bottom of the ocean. And just to show a regional assessment, and this is a very interesting paper, recent paper, showing that, okay, we have the top predators, right? The sharks and the top predators, and they rely on the fish and on the megafauna, on the big benthic animals, invertebrates, eating on the seafloor. So as you increase depth here, you see that as most of the benthic or bentopelagic fish, they rely on benthic biomass, on benthic food. So benthos is not only important for sediment process, but it's also food for a lot of fishes, a lot of important fisheries in the world, right? So the fish eat benthos when these are the diavertical migrating communities, the zooplankton, they migrate during the day. And a lot of times when they migrate and when they meet some margin, oceanic margin, and this is between 500 and 1,000 meters depth, they eat a lot of benthic animals, and that's the white stuff here. But if you have the mesopelagic zones on the open ocean, where there's no continental margin, of course, most of these fish, they rely on pelagic organic material. So that's why you have most of the biomass of fish closer to the margins. It's not only because of production, it's because when they are closer to the margin, they have access to more food. They have access to the big benthic animals, whereas if they go to the middle of the ocean basins, in the middle of the oligotrophic gyres, a lot of them are going to be relying mostly on pelagic or on jellies, on other mesopelagic stuff, zooplankton and stuff, salps and that kind of stuff. Right. So this is really interesting. So there's a really strong link between the pelagic, the mesopelagic, and the benthic compartments in the biota on the ocean margins. And these are important processes to understand. But also, like the biological pump, and people have said this before me, is not only organic carbon pump. It also exports inorganic carbon, right? So like you've seen so far already, you see here the poles, and then you have sinking water, right? Sinking of very cold, dense water on the poles. And this water is exchanging carbonic gas with the atmosphere. So it sinks, it brings carbon and sinks it to the bottom of the ocean. And that's a sequestration mechanism, physical mechanism, right? CO2 gets dissolved with water and then sinks, and some of it gets sequestered. And that's how the ocean is in a pretty good balance. The amount of carbon in the ocean is in pretty good balance with the amount of CO2 in the atmosphere. That's the physical or chemical pump, right? And the other way of carbon getting into the ocean is by biological productivity, right? A lot of the plankton, they calcify, and so as they calcify, they sink. Microbial decomposition, right, gets the carbon out of the shells, and they dissolve, and some of that carbon gets then to the inorganic or the organic carbon pool. So the biological pump also helps the ocean to keep in equilibrium with the atmosphere, the amount of CO2, amount of carbon to the atmosphere. And I think it takes about, I don't remember quite the number now, but it's very fast, right? It's under the year. So it's about a year, you have the same, the amount of carbon, CO2 you have in the atmosphere, you're gonna have it in the ocean. So it's pretty close. So microbes also mediate the changes in ocean chemistry. So as you go, and these illustrate like different alkalinity in the ocean depths. So as you go deep, the aragonite saturation, which is the balance between pH and dissolution and formation of carbonates, it goes shallower, right? So at the bottom of the cold waters, as you have more carbon, it's more difficult for organisms to calcify, right? So so the biological pump also has an important role at the chemistry, of course, of the oceans. So what about biodiversity? So how is the biodiversity linked to this process that I just mentioned? And it is a little bit. And on Earth, on land, there are pretty latitudinal declines of biodiversity. So basically you have higher biodiversity in tropical continents, in tropical land, on land, than on the poles. So you have a higher species richness in the tropics, and the species richness decline as you move to the temperate and to the polar regions, right? That's true on land. But on the ocean, it's not that true. It's not uniform, right? It's not clear, these patterns. And you see here, this is a really recent study, 2010. These guys got all animals, not all animals, about 3,500 animals. Most of them are fish. And there's a lot of deep sea biodiversity that is not in here. But that's where you have some constraints on your data, but that shouldn't keep you to not do the job, right? You do with what you got. So, and these guys were analyzing where are the marine biodiversity patterns globally. And as you can see here, we do have very different richness, right? Biodiversity between ocean basins, right? And not a surprise, in the Indo-Pacific, that's a huge biodiversity, right? The corals and the fishes and everything else. So the latitudinal gradients, if you had a strong latitudinal biodiversity gradient in the oceans, you'd have most of the biodiversity here near the tropics, which is not true. Some subtropical areas are very diverse, including in Africa and tropical Asia. But you do see different patterns between coastal and oceanic diversity. And that's interesting. So that suggests that coastal taxa, coastal biodiversity have different patterns, different controls on biodiversity if compared to the oceanic taxa. And when we look at the coastal taxa, you do have like boundary currents, some boundary regions, and you have subtropical maxima like in the Caribbean and a lot of the Indo-Pacific. There's a huge biodiversity near the coast, right? And boundary currents too. And whereas in some areas like in the Atlantic, it's not very clear. So at some coastal sites, you do have higher biodiversity in tropical areas, and whereas in other coastal sites, you have not. So, which means that there are regional controls on biodiversity. For example, habitat. For example, if you have coral reefs, developed coral reefs, you have more habitat. If you have expanded continental shelves, larger habitat areas, that's important for coastal biodiversity. So then you don't have clear latitudinal clines. Whereas in the ocean, when you look here, the gradients are a little bit stronger, but also very regionally. And then you have very strong biodiversity in areas where you have high production. And this is a little bit associated, so it's a bit associated with the upwelling regions. So there's, for some areas, the amount of energy, amount of production, and also the production of habitats, the front systems, right? When you have cold fronts coming in on oceanic basins, you do have higher biodiversity. And these guys, when they did their statistical analysis, they found that surface temperature is a good prediction, global prediction of biodiversity. And that's really interesting. And then you think, well, what's temperature got to do with biodiversity, right? I mean, it makes more sense to me, at least, that the amount of production would sustain. It's one of the hypotheses for biodiversity. You have several, and people will keep arguing about that in the next 50 years. And it makes sense for, if you have productivity, you can sustain larger populations, and then you have the opportunity for a higher number of species, right, within that population. But that's not the case. That does not seem to be the case in the oceans. So when you look at temperature, and that's what they got their significant results, and they talk about it in the paper, the temperature ecologically may have a really important role on the speed of evolutionary rates. So when you think, we have to think that the ocean is not, it's not only 10,000 years old, it's millions of years old, right? So you have to think that throughout the ages of Earth, higher temperatures in the tropics may have sustained higher diversification events, higher evolutionary rates. And that's their hypothesis, of course, for the higher diversity, the little bit higher diversity, and the relationship with biodiversity and temperature. That's interesting. But of course, it's not sufficient to explain everything, at least these global patterns. So why the heck we have all these species in the Indo-Pacific? It's interesting, right? What's in there? I want to go there. And this is just more results from this same paper, and which is really interesting. So that's what I'm showing you. So here you have temperature at oceanic and Altaxa and coastal. And then you see that oceanic, so this is temperature here on the x-axis, and on the y is diversity. So you see that you have a pretty good correlation, right? I wish my data were always like this. It would be much easier to publish papers. And whereas the clines are different. Though the oceanic clines, they seem to reach an asymptote, whereas the coastal is a little bit different. This is for pinnipeds. And the other explanation for the temperature is that most animals, most tropical animals, they have temperature limitations, right? So that's another second hypothesis that come into the paper. So you have probably both higher evolutionary rates, and most of the animals are pretty happy with the tropical temperatures. Most of them are ectotherms, right? So if you have an animal, an ectotherm animal, we are endotherms, right? Not only me, but you are too. So endotherms, they can control better their temperature at polar seas, right? The mammals. But most of the animals, they don't. So it seems that probably the temperature has a little bit to do with the ectothermy or endothermy on the oceans. And this paper goes a little bit beyond, which is really interesting, which is the application of it. Well, so when they look at the hotspots of biodiversity in the world, so they look at the global hotspots, which means the global sites of really, really high biodiversity, what they find is that most of them are at medium or medium high of regions of medium high human impacts. So, and this can be like either climate or regional impacts. So this study also finds that, of course, the hotspots are in the most changed, in the most impacted areas in the global ocean, which is really interesting. I recommend you reading this paper. So this, of course, has like implications for management. We're going to talk a little bit about that later. That's a really cool one. And also, like I said, not only ecological process explain, like temperature, explain the pattern, the global patterns in diversity. So we also have probably geological events, right? So as I said, and this other paper is really cool too, they talk about the changes in, you know, the oceans, the plates, the tectonic plates, the oceans, the global continents. They were different than they were today at some point in time. And at the Eocene, and this era had about 700 to 900 ppm of CO2 in the atmosphere, which we might get there not too long, right? We were getting close. We are at 400 something, right? So at this era, we had a lot of the biodiversity, and this is based on alpha diversity of benthic forams. So these guys got deep cores, and they look at the diversity, how diversity changed in the global oceans with the geological age. And at about 40 million years before present, we had really hotspots of biodiversity on the Tethys. This was the former Tethys Sea, right? And as you had the clashing of continents, and then this hotspot then moved to the Arabian Sea at some point in the Miocene, 20 million years ago. And you see, Indo-Pacific was not that high at that time at the Eocene, but it became higher with time. So you had changes in the biodiversity hotspots during the geological ages. And that's interesting. They say that, this paper, they claim that the loss of biodiversity in the Mediterranean, which is today the Mediterranean and the Arabian Sea, was due to major geological events, the clashing of continents, right? The tectonics. So you have volcanism, you have a big mass of geological stuff that I don't understand quite well. But these events, they coincide with the declines of hotspots in the Mediterranean and in the Arabian Sea. And then you have the recent pattern of high biodiversity in the Indo-Pacific. And this is really cool, right? I mean, so at the end, temperature might have had an effect, like I said, on the biological evolutionary rates, but also you did have effects of the geological history of Earth on the current biodiversity in the oceans. So that's really cool to me. So geological events can definitely extinct a large number of species and create other hotspots with time. Maybe we have other hotspots in the future. So when we look at smaller scales, it's a little important. When we look at the regional scale, right? And a lot of the diversity, for example, on the margin of Brazil or the Caribbean or wherever you are from, if you look at this is the number of species in a region, and then you can, like I said, you can get just one ocean or some region. Declines, the number of species in that region is very correlated to the local species diversity and richness. So it means that for smaller scales, most of the diversity that you find at smaller scales is related to the regional scale of diversity, which makes sense, right? So if you have, for a reason, 100 species in your region, you cannot have higher diversity in a local than the regional diversity. It's illogical, right? So that's why understanding the global diversity is important and is applicable to the regional scale. And one reason, in the ecological, I mean, really quickly, the ecological reason for that is basically what we have the gamma diversity, and that's the regional species pool. So that's this line here. So that's the maximum diversity you can have in a region. And species like like I said, species, they are distributed across margins, across regions by larval transport or colonization events and stuff like that. And then you have some species and selection of species, right? And that's the beta diversity, which we call it species turnover. So at smaller scales, you have different species occurring at some areas. And these species, they can be selected by even habitat effects or by competition and several other effects, right? And so we have these ecological patterns, species turnover or nestedness. Nestedness is basically like you have a subpopulation of the same regional species. The species turnover is where we have our areas with change in diversity between others. Right? So these two patterns, they may co-occur, or some ecosystems may have one of these patterns responsible for the local diversity. Okay? And so when you look at the larger provinces, so now we're going to talk a little bit very quickly about the habitats and the biogeography, right? So like I said, this is again the global ocean and the global carbon that sinks to the bottom of the oceans, productivity. And when we look at the benthic biogeographic provinces, you see they also rely a lot on patterns of water masses and productivity. And like I said, most of those deep sea basins, they have never been sampled or are very, very undersampled. But people divide these areas based on different patterns of productivity and different patterns of water masses. So there's, in general, we believe that these different colors are major biogeographic provinces where you have species turnover between these large basins. If you look at the pelagic biogeographic provinces, they're much less, right? Fish swim much more, they're dispersed much more. So the pelagic provinces, you can see, they're basically latitudinal, right? They are very correlated to latitude. And benthic, it's a little bit different because fish and plankton and microbes, they disperse much more through the ocean currents. So it is interesting to look into that. And when you get pelagic and benthic animals, species, you do have a very intricate number of provinces. So this is really interesting because we do have evidence that major oceanic basins or some regional areas, like for instance, the Caribbean here, for example, or some smaller areas, they might have different species. They might have a significant species turnover. So at some point, most species, they are not dispersed a lot in the oceans. They are limited by dispersal. The average dispersal length, the average distance of dispersal of a benthic species in the deep sea is considered to be 100 kilometers. But that does not mean that we don't have cosmopolitan species, right? We do have some benthic species that are inexplicably, we don't understand the reason, in the Pacific and in the Atlantic. That might have to be with evolutionary origins. But anyway, so most of the benthic species have a very limited dispersal during their lifetime, whereas the pelagic and the fishes disperse much more. They have much more wider ranges. So from principles to practice, so if you get the biodiversity patterns, if we understand a little bit of the processes, we can try to apply that to conservation. And that's what we like, right? We do like to look at our work and see, well, I can apply that to some objective for conservation. And I'm going to give you 3 examples quickly of deep sea, of open ocean, how people use biodiversity to conserve. And this is the area, this is called the Clare-Clipperton Zone. It's in the Pacific. So for your notion, this is the Hawaiian Islands, right? So this is in the middle oligotrophic Pacific. And these blocks here are mining claims of several countries. So several international countries have mining claims. They want to mine nodules, polymetallic nodules, which are rich in rare earth elements. They want to mine this stuff, right? So they want to mine it from like 5,000 meters depth, right? Which is not an easy task. So, and that, you know already, this is called the area. It's regulated by the ONU. The ISA, International Seabed Authority, regulates the claims. And this is one of the first areas to be claimed for mining. And then a bunch of people, specialists, got together to design management for that, management actions, to design MPAs, marine protected areas for that area. So they could guarantee that if mining occurs at some point, some communities, the biodiversity of the area essentially would be properly preserved. So what they did, like I said, there's not a lot of sampling in the area, and which makes sense then, as you understand now, of processes. It makes sense to look for special habitats in the area, and these are seamounts in the area. A lot of seamounts, which are elevations from the seafloor, from at least, I think, 100 meters. So if you have a mountain inside the ocean from at least 100 meters high, it can be as much as 2,000 meters seamounts. It makes sense to protect them. They have different diversity. They have are hotspots of diversity. And these are another this is a map of the number of the abundance of polymetallic nodules, which is the mining, which is the resource that they want to mine, and these were used to predict. Also, they are correlated with some rare taxa. There's some rare organisms, forams, and benthos that depend on these nodules or have evolved with these nodules during millions of years. And this is a modeling of the nitrogen, of food, of particulate organic nitrogen in the same area. So they are basically using what we call habitat modeling or selection of ecological and significant biological habitats, for example. So you don't know exactly how many species you have there, you don't know exactly where they are, but you can use proxies, surrogates, to model the species, the distribution, and the hotspots of diversity in the area. And then, selecting these and making some calculations and putting them into special management softwares, there are a bunch of them out there, they came out with these 9 huge areas of marine protected areas. And they have a bunch. I mean, I'm not going to talk about the design of marine protected areas. You're going to have an excellent class, I believe, of that next week. But so the basic here is that they used habitat approaches to design the marine preserved areas, because you don't have a exact number of how many species, where they are, and how they are similar to each other in this region. So you can map them. You can make, like, using the precautionary approach and design MPAs on the high seas. The second case, and this is a recent paper, I think it came out this last month, and that's the Mid-Atlantic Ridge. This is Brazil, right? The Amazon, Fernando de Noronha, and islands around here. So this is the Mid-Atlantic Ridge, right? And in the Mid-Atlantic Ridge, you have hydrothermal vents, right? Some of you might have heard of it, which is essentially deep sea volcans, right? So you have hot water coming out at really high temperatures from under subsea floor processes, and these high temperature waters, they come out with a lot of metals, dissolved metals, and when they get colder, they flow out of the subsea floor, they cool down, and the minerals precipitate. That's one of the processes. So some of them are like also millions of years old, and so at some areas, it means that in some areas near the tectonic plates, you might have thick deposits of metals, and people want to mine that too, right? So in the Mid-Atlantic Ridge, which is also an international area and it is also regulated by the ISA, there's already about 30,000 square kilometers from France, from Poland and Russian, and they want to, like in these colors here, they want to mine these areas. They have asked the ISA to authorize mining. So they used a similar approach as I just said. They were trying, so they got a group together. ISA asked, hey, you guys need to develop precautionary measures for protection of biodiversity. Hot vents are really interesting. They not only have distinct biodiversity, these animals that are not found elsewhere in the oceans because they rely on chemosynthetic resources to grow and to live. But there are also other organisms which are not chemosynthetic, for example, fish, octopus. I'm not sure if you saw in the media recently, they just found a bunch of octopus growing their babies near a hot vent. This came out in the media and was really, really cool. I mean, you just have it was like a bunch of octopus sitting close to a hot tub, you know. And it's really cool. For example, some of these guys in the deep sea, they take they found a stingray, which takes after it puts its eggshell on the bottom, it can take up to 3 years to give a baby, right, to hatch. Right, so 3 years, imagine, right? The reproduction output of these species, they live in the bottom with low energy. It's really, really long. And they found these stingrays depositing some of egg nuts, I don't know the name, of the eggshells, near these hot vents. It's like, you know, you have, okay, stay warm. Maybe you're going to grow up faster, whatever. It's really cool. So these areas are really hotspots of biodiversity. And not a lot, it doesn't mean they have a lot of species, but it's unique. Some people think that life in the oceans emerged from these hydrothermal events. Anyway, so they did the same approach that I said. They were trying to develop ecosystem-based management. They tried to come up with biogeographic and ecosystem measures or proxies. So they came out with this idea that the mid-Atlantic ridge is of course, very different than the abyssal plain in the Pacific, right? Where you have ridges. You have like scars, like submarine canyons coming from the top of the mid-Atlantic ridge down to the abyssal plains. And these are important areas where flow of organic matter, flow of oxygen, flow of energy goes to the abyssal plains. So they try to come up with several and protect some priority areas. This is a hot vent. This is the Vema channel, which people believe, physical oceanographers know that it's an important conduit of water between the tropical Atlantic. And here they were launch factor too. So they came up with these designs of several MPAs. It doesn't mean that they have to be there. It's a suggestion of a number and of the size and the positions of the MPAs that they suggest. So they come up with several surrogates, several characteristics that they think is important to preserve, to set apart these from mining claims. And this is a really interesting approach because it's the first time they considered climate change on it. So this is the first time that people have designed MPA in the open ocean, and they were considering climate change effects on the bottom based on expected changes in POC flux and on pH and everything else. So it's a cool paper. It's on Science Advances. The third case is more local here. It's from offshore Brazil, and it's very common to other margins, continental margins. So we do have, of course, lots of oil, right, in the margin. And these guys here are octocorals, right, deep sea corals, which are, on the contrary of the surface corals, they are heterotrophic. They don't do photosynthesis, right? They are deep. And it's interesting that on the Brazilian margin, there's about twice the number of corals, heterotrophic corals, as the number of species in shallow waters. I don't know if you had known that. So people always say, oh, Abrolhos is rich and the species rich. But in deep sea, the number of species of deep sea corals, including scleractinians, octocorals, and other stuff, is twice the number of shallow water corals. That's something to think. So, but they're in the bottom, right? They're really deep. We don't know a lot about them. And the problem is that they are in areas where people go and suck up oil, right? Oil production. And we have a really historical poor offshore monitoring here. This is just a title of one report from an oil company, it doesn't matter who, that they did a report. These pictures are from a monitoring report of an offshore activity. And the only thing that they put in the report and they delivered to the Brazilian Ministry of Environment are those pictures. And this guy said, okay, so these are the guys before the activity, and these are the guys a few years later, the same individuals. They marked them a little bit. So they look at these pictures, they say, okay, this coral here, this is was, I don't know, 5 years ago. It's a lot like this guy. He's happy. This guy too here, oops, not so happy, but I think it's happy, he's happy. And this guy the same. Okay, he's still happy. So there's not a single data report. And that's the monitoring of most of the oil activity in the Brazilian margin, deep sea, the deep sea. So we do have a problem here, as you know, like I said, it's not exclusive to Brazil. It's common to other margins as well. So this is the evolution of the deep sea offshore activity. So this is in feet, right, not in meters. So now, in Brazil, for example, we do have oil production at over 2,000 2,000 meters depth of water depth, 2,500 meters, right? Which is really hard, right? Like in the Gulf of Mexico too, people get a lot of oil from deep waters. And this is the probability of a reported incident with increasing depth. So look, as we go deeper, which makes sense, the problem of something going wrong, it gets really high, right? But it's of course underreported, right? So that's one of the management problems we have in Brazil. So we try to, and then we do have, right? We do have examples. This is from the Gulf of Mexico, right? The Macondo oil spill in 2010. 800, there's a poster outside there. I just saw it. So 800 million gallons, if I'm not wrong, of oil were released in the oceans. Another 8 million gallons of dispersant, which they used to sink the oil, and were as bad for the corals as the oil. There's a lot of papers about it. I'm not going to talk a lot about it. I don't have time. But anyway, so it's pretty bad, right? We don't want to see these things happening. And there's a lot of these offshore oil rigs out there. And this is just a picture, a satellite picture of the extension where the oil can go in an accident like that, right? So you had up to 60 days of oil on the surface, imagining on the bottom. So it's a really bad accident. So we need to have better management for that. And this is the picture of Brazil. So São Paulo is here. We are here. And all of these are offshore leasing blocks. So you see, they are basically everywhere on the deep margin, everywhere. So the government leases the areas. They don't look at the biodiversity. They don't look at the animals that are there. They just lease. And then after they lease, the companies go there and make an assessment and see what's in there. And they try to avoid the corals and the stuff, the important biodiversity. But there's no basin-wide management for that. So it's different than having one oil rig out there as having 200, right? And some of these basins, some of these areas have hundreds of oil rigs offshore. And this is a model of deep sea corals of Brazil. This is from a really good dissertation from here, from USP. This is for Octocoralia. And then you see the green areas. So where the corals where the best habitat for octocorals and also for scleractinians, which are the reef builders, right, is right on the margin. It's right between 200, 400, and 800 meters. And that's where the oil rigs are. That's where the oil stuff is. So it's really a problem. So we tried to do a study. So we came up, I had a student from IBAMA, which is the Ministry of Environment, and he came up with this idea. And I said, okay, let's do this. And then we got, this is Campos Basin. Rio de Janeiro is right here, right? So it's near Rio, a little bit north of Rio. And Campos Basin, these are the oil offshore leased areas. Campos Basin is where about 70% of Brazil's oil comes from. 70% of our oil is produced there. Most, of course, in the deep sea. The deep production of oil has increased by 1,000% in the last 5 years, 10 years, whereas the continental production has decreased by 90%. So most of the oil comes from deep sea basins, right? So we said, well, there's a huge overlap. So only in Campos Basin, there's over 11,000 kilometers of leased areas. And in some areas, 46% of the leased areas overlap with vulnerable deep sea ecosystems. Some ecosystems, for example, cold water corals, from 500 to 1,500 meters, some areas are almost entirely within leased areas, which means we can't even access them to study. So most of the deep sea corals in Campos Basin are within these blocks here. At least the corals that we know where they are, right? Like I said, I mean, we know the limitations of the data, but the corals that we know there are, where there's evidence, physical evidence, not bodily, but the physical evidence, almost over 50% of them are inside oil leased blocks, right? So we tried to make the spatial planning for that, and that was the first time we have done that in the South Atlantic. And then we came, like I said, with habitat and planning approach. So we use depth as one surrogate. We know that depth, there's an important biogeographic range of species, and they are deep sea species. They change, the assemblies change with depth. So we use depth as one surrogate. And these are the maps of deep sea corals, submarine canyons, and the seamount in the area. We also use sediments, the different sediments, sand, gravel, and sediment organic carbon, like I said, food. And we also use the diversity, right? This is benthic diversity in the same area. So we use these surrogates to make a special planning approach. We use this software, Markson. Maybe some of you have worked with it. It's a spatial planning approach. So we told Markson to develop a few scenarios for protection. And in this first scenario, we did not consider the current activity. So I say, hey, pick whatever the most important areas. And of course, and hatched areas here are the current listed areas. So Markson picks the important areas based only on the data we gave him. And a lot of these areas, of course, overlap with the oil rigs, the current oil rigs. So that's not politically viable, right? And then we made different scenarios until we get, okay, let's try to avoid the listed areas as most as we can. So you're going to increase the cost of the software of getting these listed areas into the planning approach, into the preservation areas. So we can have at a minimum chance of selling it to the government or to the oil companies, right? So we didn't want to design, so this is the result. So these are with all the limitations, and these blue dots here are where the oil rigs are, and a buffer zone of about 5 kilometers. The 5 kilometers comes from data where we see that most of the impacts from oil rigs is restricted to a radius of 5 kilometers. So we want to avoid that because probably it has some interference, and there's safety measures as well. So we just said, okay, let's avoid these oil rigs that are there. And you see that some blocks, they are pretty much empty, right? Which is an advantage. So the government sells a huge area, but of course the oil rig is going to be a spot or a few spots in that area because they don't need to drill the whole thing, right? They just need one pipe down there. So these are selected areas based on changes in fauna, in biogeography, in energy, and in diversity. We didn't want to design it because it's not our work to make. This is for the Brazilian Ministry of Environment. So basically, we said, okay, we can design this approach, we can come with an approach, even though we don't have enough data, biodiversity data, and we can come to an approach to use some of the processes we know, some of the patterns we know to make management happen, right? So a summary for this first part is basically, so we do know many of environmental and historical, right, even geological gradients and key drivers of biodiversity. So if you know the global stuff, you can try to apply it to your local study sites, to your local case, to your local question, and think, well, what's important in my study? What's important for me to consider? Is temperature important? Is production important? Is currents, right? Sun, etc. So if you read broadly, and I always tell my students that, don't read only the stuff about your work, read broad papers, right? They're going to expand your horizon, right? It's good for you. And you can try to apply in your subject, in your project. We do have, like you're listening to this the whole week, we do have many local, regional, and global threats. And you can also probably identify these for your case, right? So you know what's going on in your work and how that affects diversity. So in a short budget scenario, there's also been talk about it in the first day, right? We don't have money to do everything. That's the truth. It doesn't matter the economic situation. You never have enough money as you want, never. But how can you use other data? How can you use secondary data? How can you get data? And today we do have internet, which is such a powerful tool for data, right? I've seen some posters there. People use Ocean OBS, right, which is a huge biodiversity portal globally. You can get data from that. There are portals such as zoological portals, worms, and all kinds of stuff. So there's a lot of data in the internet. And data, fortunately, is getting more and more open access, right? So this paper that we just published about, the data is there. You can get it. You can get even the map and do whatever you want and say, hey, these guys are crazy. Just do something else. You can expand on that, right? That's science. So, I've learned that since I became a professor. So, like I said, I was a deep sea ecologist, and if I only had written deep sea proposals, I would never make research again. I would never get a single data again because we don't have money for deep sea, right? And as you now don't have money for many things, it's not new. Scientists, at least Brazilian, and even in the U.S., right? The U.S. is a rich country. I'm just getting U.S. as random, right? You can get any European and other rich country. Approving a grant, it's such a hard doing. The success rate is like 2%, 5%. I've known many early career scientists that they battle for 2, 3, 5 years until they get their first grant. So even in countries, even in times where we have money, or the country apparently has money, there's never gonna go enough, that we're never gonna get happy with that amount, the amount of money going to science. So you do need to, and of course we cannot wait there to apply it, right? To conserve, to propose new things, and like you're doing now, to do your job. So you gotta be creative, right? You gotta be moving around and and trying to come with approaches to do your research. This is just not it's not discouraging. I'm trying to encourage you to look for new ways of getting funded, or getting data, or getting stuff done. Okay? Okay, I'll stop now and we can have a few questions, I think. If you wanna, Hello.
Hi, I'm Mohammed from Brazil and Pakistan at the same time. Thank you for your awesome talk. I would like to know about the proxies you use for getting the biodiversity of the deep seas. And also I would like to know about, do you use other proxies for paleobiodiversity? Thank you.
Good question. So in our study case, we had a few samples, right? So it's very hard to estimate diversity if you don't have a single sample from an area. But like I said, in some areas, like in the Pacific, people didn't have enough samples to estimate biodiversity. In fact, I don't even believe that they use a lot of biodiversity, which means richness or benthic diversity. So for the deep sea, as you don't have a lot of variables, like I said, oxygen is pretty much constant, temperature is constant, you have variable flux rates, productivity. That's a pretty good way of modeling, of using that variable into your predictive diversity. So you're using the precautionary approach. You're preserving areas where you have more energy. And also, as you know, substrate, right? If you have nodules in your case, right? Or if you have massive sulfides, you can use maybe some geological proxy to say, okay, I'm gonna not use biodiversity, but I think there's some reasonable thinking to look to protect a part of that habitat that has larger sulfides, where the sulfide is thicker, and also getting areas with thinner areas. So it's basically like by habitat, and we have done that before. So like I said, if you have different habitats, even if this habitat is like a sand bottom, mud bottom, or massive sulfides, you can say, okay, each of these habitat has probably different biodiversity, so I want to protect 30, 50% of that specific habitat. That's where it's mostly done, because yeah, for deep sea, you're not going to have enough data. What was your second question, sorry?
Do you use for paleodiversity?
Paleodiversity. I have never used, but I don't remember now. There might be studies out there that I have never used paleodiversity. But like I said, it can make sense, right? Forums and stuff, it can make sense. So.
Hi, I'm Avi from Sri Lanka. I'm just curious on your opinion about how war activities can affect the marine biodiversity. Actually, why I'm asking this question, I should tell you why. My country had been exposed to a 30-year civil war, mostly in the northern part and the northeastern part of the country. And at the end of the war, luckily we ended it, we found out that most of our coral reef of the northern and the northeastern coast had been entirely damaged. So what we did, what we wanted to do, what we actually tried was, one of our leading cement companies, they made cement bricks, artificial cement bricks, and put these bricks in the water to try to reestablish the same environment, but it appeared that we could not achieve the same natural heterogeneity anymore. So now, okay, we have an artificial coral reef, but it's totally different, and it does not seem like what we have in our southern coast. So this is why I'm asking this question from you. I mean, I would like an answer with some recent examples from the world.
Cool, great question. I'm going to talk a little bit about that in the next part, but what people have done, like corals, they have larvae, right? So you know that probably. So they colonize a little bit by larvae. And so a lot of people have done that before. Coral restoration work is being done. I think there's a poster out there about that too. And it makes sense for you to predict, to estimate the dispersal if you can, or try to come up with a set of areas for coral dispersion so they get recolonizing, right? It's going to take time, of course, right? It's not something that you can just go there and plant, right? So it makes sense for you. Where people have been done that successfully, they come up with a set of, a set of really good variables that explain coral diversity. So if you go to the natural areas, the preserved areas in your margin, in your area, you can look what are good predictors of biodiversity in that area, which is healthy, and try to select similar, I know they're not equal now, right? But try to select similar oceanographic or similar variables that can foster in the future healthy corals.
But do you think that it will reach to the same heterogeneity? Can it happen? I mean, the same heterogeneity, the natural heterogeneity?
I don't know. I can predict the future, right? The future. So it's like I said, it's an approach. It's a restoration approach. So you probably, maybe you will never reach the same diversity as natural areas. So, but if you want to foster corals or living organisms to get and recolonize that slowly, it's an approach to do.
And do you have any idea on any other examples from the world?
Nope. But there are papers out there. So
right. Thank you.
Google it.
Anybody else?
Hi, Rafael from Brazil. As you are a professor at Espírito Santo State, I would like to hear from you about the dimensions of the impact on the benthic of the Fundão disaster, something around that.
Great. So just to quickly, right, we can talk about this for hours. So we had a mine tailing accident in Brazil in the Rio Doce, in a river. A lot of mine tailings went down river and reached the coast. That was in 2015. And we've been studying, including Luis here, my student, he has worked with that. You can talk to him about that too. The initial impacts we saw, like typical impacts, lower diversity, a lot of metals accumulating in the estuary and on the nearby coastal zone. You had burial effects on the fauna, and since then, since 2015, we got a project from the government to monitor it in the estuary, right? So we're doing it, and we've been doing that for 2 years now. So a summary is that, like in the estuary, heavy metals are accumulating, and sometimes they come down because there's a seasonal flux, right, variation. So during the dry periods, we have really high cadmium and other heavy metals accumulating in the bottom of the estuary, and that's going to the fish. So you have contamination of fish, invertebrates. We are seeing initial results on that. On the coast, I haven't seen a lot of data. There's several people are studying there. Probably 30% of the Brazilian scientific community is studying that accident now. So the good data that I have seen a lot of data. Some of that are good. And we do have the position on areas that, mud naturally deposits near on the continental shelf. And those areas are probably also accumulating some of the metals. And we can expect to see some changes. And I believe that's a personal opinion. I haven't seen data again, that most of the damage is going to be restricted to the nearby the estuary, nearby the accident. So we do have some, you have seen probably in the media, sediment plumes going to the Abrolhos or to even 200 kilometers south. But we don't have any evidence really that there's accumulation of metals. And that was a problem at the beginning. People were just scared to death. That some people were like, oh my gosh, Abrolhos is gonna, all the corals are gonna die. And so I think most of the impacts are gonna be restricted to shelf areas nearby. But even then, we do have, we do have rhodolite beds, corals nearby that can be affected. But I don't believe that the damage is gonna be, for example, I don't know, I'm just kicking up a number, like over 100 kilometers away from that, from the Rio Doce. I believe that the ocean, like I said, the ocean is pretty big. So the material that gets into the continental shelf, it gets, it just spreads out. And there's a lot of wave action on the coast, tidal action, tidal transport. A lot of that is going to be slumping down to the deep sea. There are submarine canyons that can take stuff to the deep sea. I would like to see that too. But it's everything now. We don't have any data. I haven't seen any data on the strong data unequivocally showing impacts from that accident to marine organisms on the shelf, on the marine. But on the estuary, we do have data showing that there's an impact, there's metals that are accumulating, and that's going to the fish and some other animals.
Hi, my name is Raibel from Venezuela. So I have to quickly question, I guess. In the first part of the presentation, you talk about the original biodiversity, but I was thinking about when you refer about the original biodiversity, you are comparing between eras, or okay, it's just to know about that, because maybe I hear sometimes when I was reading about all the evolution of Earth can just try to make a process about diversity of some animals and can get more species in different part of the oceans. So how can you estimate that original diversity when you don't you have a different process of evolution in that part?
Great question. Great question. The estimated geological diversity, right? People, I mean, you cannot know what were the ecological processes back then, right? The oceans were very distinct, like I showed. So you had much higher CO2 concentrations in the atmosphere. You had different temperatures. You have different oxygenation, circulation patterns. Most of the things were different. And what they have done in that paper specifically, they just get a number of foraminifers in cores, and they were just comparing, okay, here we have more forams at this strata at that age. And some of these forams, like Edmo said, they moved some of these lineages, right? Some of the same, even if not the same species, some of the same lineages, they moved around different hotspots during the geological era. So they're just counting the number of animals at some specific time.
Okay.
That's the most of the geological approach.
Okay.
And they expect that makes sense, right? So.
Okay, thanks. And the other question is about the whole exploration that some industry of oil and gas that made in different part of the ocean. They are just dredging the sea bottom in some places. For example, in Venezuela, it's too common that, we are an oil country, so they are just dredging the sea bottom, and they found a lot of species that right now, I think that researchers, they really don't know what is the role of that species there. So I really don't know if there are some regulation maybe at the world, or if that depends of the country, about that animals that maybe had a big role in that kind of those deep ecosystem. For example, corals, they found a different species of coral sponge, and that a lot of animals there. So I really don't know if there are some knowledge about that kind of things.
So most of the impacts of the oil are restricted. Like I said, they are really mostly of the drilling is restricted to like about 500 meters radius, typically we say 2 kilometers radius. So regular operations typically impact a restricted area near the oil rig, right? So they drill and they impact nearby. And the trawling, which I think you mentioned, the deep sea trawling, the fisheries, in Brazil, they don't occur near oil rigs because of safety measures. So in the most dense oil production areas, people cannot trawl. And in Brazil, there's not a lot of trawling because it's a low productivity margin. It has happened before, up to the 2010s, but nowadays it's not very productive, so there's a lot of deep sea trawling, right, which is pretty damaging. It's much damaging than oil. Concerning species, every time you sample below 500, 1,000 meters, you're going to find new species. That's a pretty common pattern. So there's several species we don't know. And recent data, including there's papers published recently that showed that the oil rigs can have benefits for diversity because they are artificial reefs, right? So in the northern ocean near UK, the northern Scotia and stuff, they have been drilling oil in the deep sea for decades, and they see that several species of corals, they settle on the oil rigs, on the structures, and these oil rigs now they work as stepping stones for these corals to disperse. So they have some benefits for dispersal of organisms, like I said. So they may be used as conservation measures. They increase connectivity, but I think that was the question. So there's always new species. In Brazil, we also don't know how many species, but only the species that have been identified, we know there's a lot. So.
Thank you, everyone. I think we are over the time, so coffee served. You can leave your question for the second part with the Professor Angelo. Okay? Thank you.